I would have started out with zombie deer. Organelles. We were finishing organelles yesterday. We didn't quite get to the end of it because I didn't want to rush through it. Somebody asked me a great question walking in, and they said, when we went over the smooth and rough endoplasmic reticulum, I always try to pass on questions that I get in passing to the rest of the class so you think the same way. And the question was, well, you didn't really tell us a lot what smooth did. We talked about what the rough does, immature proteins in the Golgi apparatus and the Golgi complex. And that's going to be that way with a lot of things in anatomy. Right? Some of us had anatomy before we got here, like I always say, but now we're adding to that. And if we were to really dive deep into every single little piece we went along, we wouldn't get to where we're going. We wouldn't get to what we need for PA school. Like I said in the beginning of the first day, that's my job to kind of delineate that and figure out what we need to know. So some things like smooth endoplasmic reticulum didn't have anything there that we talked about. If it didn't, we're not testing it. Right? Now, you need to always ask those types of questions to instructors around here, especially if, remember, I went through this program. You get into cardiology and you gloss over something like that, and then you get to the test and they ask about it. You know, that's why I understand this question that was asked this morning, because they put it on there, but then I expected you to go read that. You know, it was in the reading assignment. Right? Not for me. I'm trying to help you out, not for me. If it's not on that slide, we didn't discuss it for a reason, and I don't expect you to know it. Fair enough? That's the whole reason I passed on the question they asked me. It will be on the slide. I only give 50 questions. Each test, we have 7 tests, I get 50 questions. Look how much info we have. So, in my mind, I always do it, and I always hope this helps y'all when it comes to testing. It would help me if I was doing it, because I always say this. If it's important enough, if I only get 50 questions, if it's important enough to put on a test, then it's important enough to at least mention in class. It's important enough to discuss sometimes, and it's definitely important enough that it better have made the slides that I'm presenting. You see that kind of attitude? Now, sometimes you look at a question and go, oh, that's kind of small data. Okay, but it's there, right? We did talk about it at some point. There's never going to be a situation where you get a question and went, oh, if I'd have done the reading assignment, I'd have seen that. Okay? Fair enough? That's the way I'm playing the game here. Organelles. We're down to the last ones. Lysosomes, peroxisomes, proteasomes. Lysosomes, we already mentioned, we just didn't talk about. We said they're breaking down like bacteria and things that comes in. There are digestive enzymes that are found inside of the cell in vesicles. So you bring something in through endocytosis, it's going to combine with a vesicle, right, that has lysosomes in it, and they can digest that and break those products down. Right? Now I mentioned it, they're formed from the Golgi complex, but mostly everything goes through the Golgi complex to get formed, right? That's where we mature proteins, where we make things. So here's all your official statement on a slide. They digest things that enter the cell, but they can also do autophagy. They can break down organelles. Organelles inside that cell will wear out. They can break those down. They can also do autolysis, which is they can basically digest the entire cell. Sometimes that happens. The body has a lot of different processes that go on, but for now we're just naming what lysosomes do and what's the difference between autophagy, autolysis. Okay, we also have peroxisomes. Peroxisomes has perox in it. One of the things it does is it helps to break down hydrogen peroxide. Trying to get my pen going here. Hydrogen peroxide. So when you go through like the Krebs cycle, oxidation cycles, that kind of stuff, right? Byproducts, it can help to break these down. Hydrogen peroxide. And you find a lot of them in the liver, but another big thing I would say they do is they detoxify substances such as alcohol. So when you drink, they're going to help break that down as it goes through your liver. And then the other one we see is proteasomes. There's actually a lot of study about proteasomes, even more recently, because there are a lot of neuromuscular type diseases and things, and Alzheimer's and all this, where you get like abnormal protein buildup inside and outside the cell. And some of it's related to, they think, to proteasomes, because when you look at a proteasome, its job is to destroy, degrade, break down unneeded, damaged, or faulty proteins. So if you have an abnormal protein that's made, because things don't go all the time, things do not always go like they should during protein synthesis. So if that protein's abnormal, these proteasomes are there to break it down and reuse those products. But if you have a fault in this area, that could lead to certain things where proteins accumulate, like in the central nervous system. Now, obviously, I'm getting outside of the slide, right? When I start spewing outside of the slide, giving good information, it's not something that you got to write down and try to remember. Right? Found in the cytosol and nucleus, because I was thinking that going, I spent time talking about it because it makes it interesting, but really all you need to know is proteasomes break down proteins. I know, some of y'all are like, well, just say that. That's not as fun. It's not as fun. Right? It's not as memorable. All right, vacuoles. We don't need to know a whole lot about vacuoles other than basically a vacuole is a larger vesicle. Now there's more to it than that, obviously, but vacuoles are a very large type vesicle because they can be associated with lysosomes being in there. You can use vacuoles to store some materials that are in the cell. Right? They're just like vesicles, they're formed from part of the plasma membrane, either a Golgi body, the plasma membrane, however it's made. But just think of them as a large vesicle. And as we near the end here, you have the centrosome. All right. The centrosome is associated with the microtubule organizing center. When we talked, this links into, when we talked about the cytoskeleton of a cell, the cytoskeleton had microfilaments, intermediate filaments, right? And then these microtubules. And so where these are actually made in the centrosome here, it's going to assemble these microtubules, and you have these 9 triplets here of tubules. So basically all you need to know about the centrosome is it is the microtubule organizing center. And we're going to send those out from there. And then lastly, when we talk about organelles, we have cilia and flagella. Cilia are short hair-like projections that we find along the surface of some cells. Not all cells have cilia. If you look at the small intestine, it has cilia, microvilli. They're generally helping to move things along the cell. Right? That's why the bowels is a good example, because food is moving through. We want to keep that moving through. We also want to be able to digest that food. So cilia move things along the cell. And then flagella, you only find in one place, and that's a sperm cell. Right? It's a longer cilia, and it actually does like an undulation or this motion over here to propel a sperm forward. But when you look at a cilia, it kind of does a wave-like motion. Right? It kind of goes back and forth, and then if you've ever seen a sperm cell video, right? It's, I don't know, I call that undulating. Did you think about a sound wave? All right, that's all of your official organelles. But like they do in a lot of things in anatomy back when they did this, when they used to dig up bodies in secret and all this. Yeah, this came to my mind too. Sometimes they got lazy though. You get to Friday, you're trying to name all these organelles, and you get down to the end and you go, all right, let's just call the rest inclusions and call it a day. That's not really true, of course. Inclusions, when we go back to, and we said the cell is membrane bound, organelles have their own membrane. Remember, like the Golgi complex has a membrane. Well, most organelles in a cell have their own membrane also. But there are some things found within the cytoplasm or cytosol of the cell that are not membrane bound, but they're in the cell or stored in the cell. That would be called an inclusion. So glycogen, lipids, pigments when we look at some tissues, crystals, all of those might be found in the cell, but just not membrane bound. That's the straight definition. And then we get to the definitions. So I don't go over the definitions in class, but I don't know if I told y'all this, as the years went by that I've been here. You know, I think I came back here, I was here from 2010 to 2013, teaching AMP2 on active duty. Then I came back in 2016, and I've been here ever since. And so as things progress along, and I come up with an instance where somebody didn't know what something meant, like it was asked somewhere else, I just happened to hear it, or it was on my test, I try to throw it into a definition so that we know that, right? Remember when I talked about instructor bias, like my friends know what the apex is. And so I'll add those things in so that you know too now. But because I do that, on every test I generally do go and ask a straight definition of it. So you need to go through them and make sure you know what they mean. You don't need to know if it, you know, those are not great examples, but malignancy is. So malignancy, cancerous neoplasm, you need to know the straight definition. You don't need to be going looking up what causes malignancy. Don't go read anything else about them. Just y'all know how to do definitions, right? Learn the straight definitions. Don't get on sidetrack about what they do, what they mean. Don't go look up leukemia. Right? Just know the definition. Because some of these we will talk about again. Others will be other classes. But they fall kind of funny now because I used to have all the blocks separated, as in genetics, neuro, each one had its own block. But now all of these are combined into one because of student preference. Right? As the internet sped up, the students wanted them all combined into one. Now why they don't do that themselves, I've never understood. It's just like, I'm going to throw this out there. This is one of those things, you know, sometimes you offend people unknowingly. But I get these back and forths all the time about the slides and colors on the slide. People are like, well, I use notability, I don't want the color on the slide. Okay, great. The next person next to you goes, keep the color. I need something when we're talking. I can't stand straight white slides. It drives them crazy. Just like if you were to ask everybody in this room, should we move the AMP2 test to this Friday? Some of you would say yes. Some of you, you'll never get an agreement in this classroom. I know it. I've been around a while. So I've gone back and forth. Colorful slides, the thing, remember the old PowerPoint things flying in everywhere people used to do? You know, I never really liked that, but I used to have some pop-ups, but the pop-ups, when you start pulling it out into other PDFs and stuff, they cover stuff up then. I got rid of all that. Anyway, so I got a compromise going on. You notice that? They're white, but a little bit I like blues. The point here is, I've never understood. You're more technical than I am with computers and phones and stuff nowadays. I'm starting to admit that as I get older. It used to be I'd say, no, you're not. But now I'm thinking you are. Change the damn color yourself if you don't like it before you put it in your ability. Get in the notes view or whatever, go control. Oh, you gonna throw a wrench in that?
Oh no, I was just gonna say they had to mix stuff up. Whoa.
That's good.
You said that from the position.
Yeah, so I saw it on right? I didn't think about that till last semester, and I went and asked my wife actually. I said, they're always complaining about everybody's slide colors in PowerPoint. I'm like, why don't they change it themselves before they put it in Notability? She's always like lawyerish. Just, well, maybe they don't have PowerPoint. I'm like, and you can still do it in free Google. There's free PowerPoint. You can do it. You know how to pull it into Notability and all those other crazy apps y'all use. You can definitely know how to change the background. So I'm sticking with this right here for the next 3 years. Or at least tomorrow. Because every day when I hit the stairs, I make a decision. Am I going to go on in or am I turning around? So you never know. People ask me all the time, I hate the question, how much longer do you have till you retire? I'm like, it could be tomorrow. I don't know. It could be this afternoon. I'm not sure. It could be 10 years. I'm still having fun. It makes me think they're trying to get rid of me. Okay, it may be. Who knows? I can't worry about that. Genetics. Genetics is fun, but we can't get too off track in genetics. You're going to want to, in the backside of, in the beginning of genetics, you're going to be like, the hell is this stuff? In the end, we're going to talk about some disorders, chromosome disorders. That gets a little more fun. But we got to get through some basic stuff. I like genetics to an extent because there's a lot of studies going on and genetics is kind of interesting. But if you look at genetics, it's just, or you're a geneticist, right? You're going to study genes. And it's what one of my daughters is going to school for right now. She goes to A&M, but she went there thinking she was going to do forensic science, and she got into classrooms, there's 900 people, and she's not that type, she's real talkative, unlike me, and she wants to interact with people and get to know people. Every day she comes in, she's sitting by somebody I don't know. Anyway, she don't like A&M, so she is transferring to Sam Houston State, which has one of the best forensic programs in the nation. And unlike A&M, who you got to go to school for 2 years before they accept you into forensics, Sam Houston accepts you the first time. She's already accepted into the forensic program. So I'm saying that because it's weird that I talk about all these crime shows and I got these people. She's going into forensics because she wants to have a double major in genetics. And I'm like, I'm glad you like math and science because that's a tough. I looked at her course load. I'm like, holy crap. Anybody do genetics? Forensic genetics is a lot. I mean, you got to be dedicated. I don't say you got to be smart. I don't believe you have to be smart to pass everything in the world. I think you got to want to do it. You want to do it, you will. Okay, sorry. I'm going to stay on track the rest of the hour. Genetics. My kids aren't jealous concerned. I may talk about my son more this semester. Because he's been the topic the last 2 semesters. The other classes even ask me how he's doing. This dude's 23 years old, and he's in that phase where there is no way that anything a parent says could be the right thing to do. You know that phase. I went through it too. Dude calls me like, I said I was going to. I watched. Called me last night. And I said, hey, did you overdrew your account again? Because you haven't been working. And he said, yeah, because, you know, I have a who works oil field work. You know, you make like $3,500 a week, but that leads me to what I told him. And he said, yeah, but we had a guy, a boss, that ran this team, and he lost the job for us because he went off on the guy we were at, so we've been out for 3 weeks and haven't gotten paid. And I said, so that's why you overdrew your account? And he said, yeah. I said, bullshit, sorry. They quit cussing me. BS. He said, well, what do you mean? I said, that's BS. You make $3,500 a week when you're working. I get it, it's sporadic. I said, but people think that way. Well, the boss lost the job for us, so it's his fault. No, I've been telling you for the last year, you should have money backed up for when that comes. You're in oil field work. It's not going to be steady. Y'all agree with me or not? I said, it's not his fault. Yeah, that sucks he lost a job for you, but you overdrawing your account, it's your fault. Hopefully y'all see what I mean. My kids love me. If you have daughters, I'll tell you this. They don't want you to beat up their boyfriend, but they want to know you would if you needed to. They want you to. There's more to that, but okay, genetics. Study of genes. I'm not up here to just stand up, so I need to get on this. I need to stop. Tomorrow I'm going to try not to digress talk. I've already ruined it today. Okay, mathematical principles that show us as we evolve or as we pass on through generations, we transmit these genes from one generation to the next. Right? How we get different individuals. So a gene is just a hereditary unit. Sorry, did y'all's slide cut off like that? Thought I fixed that. It changed all the sides for it. It's a sequence of DNA on your chromosomes that codes for each little thing. Can y'all not see it at all? Even if it's resized? Let's see what's going on here. But you have it on there, it's just my slide is screwed up. probably more that are like that now than I did one. One. So a hereditary unit consists of a sequence of DNA. When you look at chromosomes, they have DNA sequence on them. And they have a specific location on that chromosome for those genes. Like blue eyes, brown eyes. They're going to be the exact, they can actually map that out and show you where your eye color, or the genes that determine your eye color, are on your chromosomes. They can figure that out. Right? They have figured that out. Wait, killing me, Smalls. There can't be that many. There was only 2 or 3 that did this last time. So let's see. I'm fixing several. Y'all will have to let me know as we go along if there's more, but there shouldn't be.
There's quite a few.
Is there? Oh, that's good. That's great. That's a positive. I will go home. All right, here's a few. Here we go. I will. When I get home today or at work, I will change it and repost it. Okay, I did that last semester too. It didn't work again. I don't know why it's doing that. But I'll fix it for you so you don't have to, and then I'll post it back up. When you look at chromosome terminology, we're going to spend a little bit of time on this side because a chromosome is the definition of a carrier of genetic information. Right? Chromosomes are normally, we said, relaxed. Remember chromatin inside the nucleus? It's all relaxed in there. It looks like cotton candy. The only time chromosomes can be represented in what we draw them as is during what's called mitosis or meiosis. And we're going to discuss both of those, but mitosis is like replicating new cells from one cell creating 2 more cells, complete replication. And meiosis is actually a reduction division for sexual reproduction, right? We're going to get more into that later, but the only time they start to look like this picture is during basically metaphase of mitosis or meiosis. If you don't know what that means, that's fine, because we're going to study that, but I'm telling you this ahead of time to show. You'll see chromosomes represented, and they'll say, okay, well, you have all these chromosomes, and you have 1 chromosome for mom, 1 chromosome for dad. Notice draw it like that. All right. Well, each one of these, when they draw that chromosome looking like that, that is actually this. Okay. All right. Most of y'all familiar with basic chromosome, right? It's a double-stranded helix of DNA, right? It's a carrier of genetic information, but it's a double-stranded DNA molecule. But what the picture is representing, and you can't see it, is where it's coming from over here as it's spiraling down, right? It's all out here in the nucleus as chromatin. And then it's condensing into this form, which it's going to do during mitosis and meiosis. We'll understand that later. For now, we just need the nomenclature, and that helps. So when you say a chromatid is when 2 double-stranded molecules of DNA are joined in the center by a centromere. All right. So each side is called a chromatid or a sister chromatid. I like to start out with this slide, and hopefully when we get to it, you really understand that. Is since they only look like this during, let's say, metaphase of mitosis, a certain phase, they line up like this because when you have 2 sister chromatids, what's going to happen is this is a chromatid. This is a chromatid. But representative, if you said, well, what would you call this whole structure? That's a chromosome. Because a chromosome is a carrier of genetic information, but they can look different ways. Is chromatin a carrier of genetic information, of course, but it's a different form, right? It's relaxed. But when you, if I was to, let's see if you're following me. If I came in here and I said, okay, well, what is this? If I circled here, what is this called? Just that left half. Chromatid. Right? What we don't know yet, and we're going to get to, and we got to link back to this, is when you go through mitosis, we need to double the amount of the DNA so each cell can have the same amount. So when they double it, you're going to take the original DNA and copy, and they're going to be connected. In that case, they're called sister chromatids, connected by a centromere for only a short period. So each side of this, you would call it chromatid. But then we're going to split those apart, right? Y'all seen cell division. When you split them apart, you're going to take the chromatids that are connected, pull them apart, and they will become chromosomes of their own. Right? That's representing this picture. I don't see in the eyes that everybody's getting it, but we'll get it. Each one of these is you would say this is a single-stranded DNA molecule. This is a single double-stranded DNA molecule. This is a single double. So it's a double-stranded on both sides, but they're an exact copy of each other. So just remember that, and then when we get into how we made that copy, it makes sense. Okay, but for now, chromosome, chromatid. Okay, we'll get to it. I don't think we were tracking there. I said it backwards or something, but that's all right. We'll get it. Autosome. Easier way to think about autosome is you have one pair of sex chromosomes. If you have a chromosome that's not a sex chromosome, it's called an autosome. Right? So it's the ordinary chromosomes. It doesn't determine sex. But sex chromosomes, we're going to have X and Y. Right? Biologically representing female and male. Okay, another definition we need here is homologous chromosomes. So these have a certain categories that we're going to see. They have the same pair, the same linear sequence of genes. They look alike, and then they're going to pair during meiosis. Okay. So in each one of you, right, unless we have any type of genetic abnormality, humans are diploid, meaning they have 2 of every chromosome. So if you look at chromosome 1, you're going to have 2 chromosomes there. And one came from mom, one came from dad. Right? It's an easier way to think about it. So biologically, you get 1 from each parent. And so you would pair them up. And when you pair them up, on chromosome 1, one from mom, one from dad, they're called homologous chromosomes. And the reason for that is they have the same linear sequence of genes, they look alike, and they're going to pair during meiosis. Right? Some of this is defining before we get there, because we haven't really talked about meiosis, but we got it. So you have a homologous pair. So we have 23 homologous pairs, 1 from each. Another terminology is locus, just defining them, the position of a gene on the chromosome or the position of a gene on a genetic map. That's its location or its locus. Then the centromere is where you take 2 sister chromatids and you connect them in the middle. The centromeres, that you're going to have spindle fibers that come off and attach those 2 chromosomes temporarily during mitosis and meiosis. So you would say that's a centromere. And then another definition, telomeres. Telomeres are interesting. I've neglected to go look this up, so I saw it. But have you seen the medications they have out there now that are supposed to prolong or keep your telomeres from disappearing? There's a lot of interesting stuff. Even like zombie, there's some conspiracy stuff about telomeres, and I'll tell you what it is in a second. So telomeres are DNA sequences that sit at the tips or the ends of chromosomes, out here the darker blue. And what they do know is that, well, they think they help the chromosomes from sticking together, because if you look at like trisomy 21, Down syndrome, you have an extra chromosome, the only way that can occur is if some were to stick together, so they think telomeres help that. But as you age, they do know this, your telomeres will shorten. Right? And since they think that telomeres, they're not really genetic type information that's determining the individual. They think telomeres protect all this DNA material that's in here. And so as you age, they've seen that those telomeres, shorten. So when your telomeres run out, I guess your cell dies and you die. That's the underlying theory there. Aging, death of cells. Now we got meds that say they're going to prolong your telomeres. Without reading it, without knowing, with no further knowledge, I'm going to call, whatever. I like the better conspiracy theory of it is apparently some people believe that aliens came along a long time ago and decided, you read different historical accounts, whether you're talking like religious accounts or you're just talking papers, things that were written on walls, that people lived for more than, what do you live, 85? It's talking about people living like 400 years and stuff, and conspiracy theorists out there believe that that did occur, but something like the aliens came along and put the telomeres there to cap us off so we could. The aliens capped our telomeres and put those there and caused it that we would naturally die by a certain age, regardless, unless you had some type of other disease. Yeah, right. Okay. It's one of those things, what's that?
Checks out. Checks out.
It checks out? Shall I look it up? Who knows? Okay. Never say never. Genetic number. Okay, let's familiarize ourselves with how we number things in genetics. They use letters to identify the amount of genetic information that is present. And so you would look, instead of using just 46 chromosomes, all that, 23, they would represent them. So when you say N is a gametic number, and it represents 23. So if you have a sperm cell or an egg cell, it would be N. And N means that that cell has 23 chromosomes. Right? The somatic number, meaning humans are diploid, so we have 2 of each of those chromosomes. Right? So we're 2N. So all cells in your body that aren't going through reproduction are 2N, meaning they have 46 chromosomes. Okay, so we now represent those numbers, and that's how they're represented. These slides are killing us. It's not like I haven't changed them in a while, other than the background color. I don't know why the font changed. I can't stand it with 8 up on slides that we used to. Alright, protein synthesis. We're not going to spend a lot of time on it, except for these next, I think, 2 slides, and I'm going to explain this to you. When you look at the process that it takes to make proteins, this is how it's going to occur. You're going to have what's called transcription. Transcription is where you have your DNA that's in the nucleus. Right? The nucleus contains all of our chromosomes, all our genes, in other words, our DNA. And so you're going to transcribe one section. You come in here and you're going to transcribe one section of the DNA and make what's called messenger RNA that can be carried out of the nucleus. It's made in the nucleus and it's moved out through the pores into the cytoplasm, because what are the two things that have to come together are the ribosomes, right? We have a small subunit, a large subunit. The ribosomes reside in the cytosol. So the messenger RNA is going to take that little snippet of your DNA out. That little snippet tells it how to build the protein that it needs. I'm going to give you a great analogy here in a second. And then it has to be translated. So translation means you're going to assemble the amino acids or the building blocks there into a new protein. Right? So translation is accomplished by ribosomes, and it normally occurs out in the cytosol, but it also occurs on the rough ER. Right? So both those places can have that. So most of the details you're going to cover in that. But I'm going to give you an analogy, a better way to think about protein synthesis. We're going to use a military field problem though. Right? So we're going out to the field, and I am the, let's just say, platoon sergeant for the medical section. All right, so we're out there. We're all set up, and I hear that I need to come over to the talk. Everybody knows what talk is. Still call it a talk. I'm getting so far out from the bin, I don't pay attention anymore. They're going to change the name on me, and I look like I'm an idiot. But going to the command tent because there's a new op order. Okay? Air Force, you know what op order is. I'm just kidding. Joke, sorry, bad joke. I'm going to the command tent out in the field because I hear there's a new op order, and it's this freaking thick. Right? So that op order represents your whole DNA. Right? But I'm the medical platoon sergeant. What section of that op order do I need to look at? What the medics need to do. The op order is order for specifically for each section what they need to accomplish to make that whole mission happen, right? So I don't need the whole stack of the op order, the DNA. I just need my part. So I'm going to flip that open and get to it. That's transcribing. I'm going to go in and they're not going to let me do this. I'm going to take pictures with my phone, when they're not looking. Click, click, click. No, I don't write that down. I'm going to take it and write it down and transcribe a portion of the op order. Or I'm going to transcribe a portion of the DNA. And now I have become the messenger RNA, right? So I'm going to take that back out, and I'm going to go back out into the field, and I'm going to find me a small subunit and a large subunit. In other words, I'm going to find me, we're going to call the small subunit the corporal. I like corporals. Corporals are always miserable, right? You ever met a corporal that wasn't upset? And then, are you an NCO or are you not? When it comes to having to do things, you are. But when it comes to the rewards of that, you're not. I see the frustration. See, guys, the large subunit, though, is the private. So I'm going to take that message out to them, and I hand it to them. And the small subunit for this corporal is going to take that and read it. In other words, it's going to translate the messenger RNA. Or he's going to translate what I transcribed back there. And it says, take your GP medium tent, tear it down, and move it 200 yards northwest at this amount. And so it's going to call the large subunit over the private, and it's going to tell the private, hey, this is what you're going to do. Right? You're going to take these amino acids, you're going to put them in this certain order. Right? So the small subunit's translating and telling the large subunit what to do. The large subunit's stacking it. Or the large subunit's the one that's moving the tent that far. Right? That's translation. That's how you build a protein. I think it's a great analogy in my view of protein synthesis. But here's the problem, is I've always skirted the line, and it always got me in trouble. Because that was an actual true thing. Move our tent. Like I could see where they wanted me to move it to. This didn't go well. So it's a GP medium. They're not that heavy. We got lots of medics. And so we, under my guidance, picked it up and walked it over there. And the sergeant major saw that. Oh my goodness. Because the whole point, you look back on life and you thought you'd be a smart. The whole point is we're out there to learn and train on how to jump, move things. So the point was to tear it all down and pack it up and move over there and unpack it again for no reason. I thought at the time. But when you look back and you go, okay, so that was some good training. That kind of. Well, we did it. We wound up doing it 2 or 3 times. We were messing with that tent for the next 3 years that Sergeant Major was there. He never let me forget that. You're gonna pick it up and walk it over there. Yeah. He liked me though. Okay, now you'll hear a lot more about protein synthesis and biochemistry. They're gonna confuse the hell out of you, but always go back to that analogy. Messenger RNA, like DNA, transcribing a picture of the top order works for you. Okay, somatic. That's actually perfect breaking point. So, let's take a break and then we'll get into mitosis.
And it's specifically the sex cells are and it's recognized as the egg or the sperm. They have you're a dog, so that your normal cell, which has
The dog just wants to chase. The chickens don't do very well when you chase them. You know, it's Texas, so unlike a lot of places, most people don't mind the places they drop it. You either have to pay or you gotta go kill bullets. Yeah, bullets have a lot, but you only know what's coming on there.
that's how it's that's how you would eat. I remember if I gave you here.
Oh yeah. It's going down. I literally see them, I'm talking like 30 plus turkeys together at a time. It's how I know turkeys can't fly so well. Yeah. I know they can go at least as high as a telephone pole. I saw it do that. But those things, they hide, but if you go down into the lower part of my property, where I have a farm down there, there's a ranch house. And you can sit on that portion now, you can hear them almost go down in this creek. But when people come out to hunt them, my brother-in-law, they never see them.
The chicken and cobra.
They migrate around like acres, not just migrate. I mean, I'll see the same group a mile down the road. There's so many of them that when you do start seeing, like I'll be driving down a mile and I'll see and I said, you know, I felt it the other day and there's like 15 miles, 4 miles, all 4 miles. I'm trying to get they think that.
Yeah, with a bow at night. Not that I do that.
Yeah.
It's an option. I shot a big 8-foot grizzly bear one time tonight.
See, I said remind me. I'll tell you what. If I don't, I don't mind. If I start off, raise your hand, I'll see you and I'll know it's really takes yourself. Appreciate that. I gotta start learning besides people just right here. I get more guys. It's like a
I'll talk about the one with the problems. So you just do that staff.
Right versus left.
every class literally. I had one class, all army. That was interesting. We had a lot more. We had a lot less feelings kids. We were hurting some feelings people. It was like living in there. That's all gonna come back. Come on, since they were recording. My IT doctor said it was good.
So my buddy, he's like, oh, I got to know on that. I can't get over. He's gonna listen. I'm like, I'll work something for you. I get this out of there. Oh, brother-in-law's name. You know what I want to do? I've seen it all the time on Facebook or some videos. Is the hog hunting from a helicopter. I don't know how much. Are you paying on the range? But fly around in a helicopter, AR. It's freaking. And it pays for all your ammo. So you get, how many rounds? Probably 1,000. I saw one on the back of a pickup truck, and they're like, smoke. And you just let them lie. You don't take anything back, do you? Take freaking Atta boy. Eagle Scout right there, always prepared. On things that I think are like the fact that they're like giving kids doing experiments on them, maybe extract it, we're gonna inject them into politics. I don't know. Like literally, deep words. I read it for a bro. So imagine throwing all these kids, putting these guys underground, somebody scared of whatever they're fighting for, getting into this crazy and extracting this adrenaline and then it makes you young or whatever. Inject me into Will Ferrell. Sorry, your buddy Will Ferrell. He's one of them. Like crazy. Right? So the thing is that far off and what is like actually happening? Kids being abducted and terrible things like that. That's happening every day. I believe that. But what if you choose to sit there and be low on it? I'm living my life. No, if I just do it, well, I don't do that. But I spend my waking hours reading it from my phone. So just sits around and listens to him be it's all great. It doesn't ever go. Is there a lot of stuff that happens to like hair? Oh, terrible. What else? It also hurt me. I swear that was like his first one. His first one that he like. That was the first one that hit. So it's like he would not let it go. Great day, man. You know the earlobe therapy thing. And they replace it with a small skin. And it is the earlobe. It's like a picture. Again, potentially, I don't know. But there's some photos and some things where he has earlobes. And then there's other ones where it's so that, you know, people who are yep. Doesn't stop.
So I don't need to learn your
It's all the same.
Well, no, he said you don't have to be really
but I'll be able to
yeah, or he'll write it. He might write it like this. You do. So he might say like, yeah, he said if I go try to trick us with it, I'll look the other one that he said was 5%. He said the others, because like
Oh, geez, I showed up one time, so. Take it off again the screen here. It's got to be no camera. And then it's too high, I can't see it. Alright, here we go. Y'all kind of took a long break. Actually, I was back there talking. I always hear these things people think, like, well, you're the one in control of the class, you just didn't stop. I got it. Somebody asked me a very important question as I walked up. What's this chain for? They said they thought it was a chain of punishment. Some student, this is an example of Medco, because instructors were complaining. I'm talking 2 or 3 semesters, we were telling them that this screen, when you're standing here, now a lot of people don't use these anymore, right? I may be the only one using them right now. And if the pin worked, it's much better. Hopefully they'll fix that. But these screens are here, you have to be able to see over them. You don't want to be behind them here. So you can't see people in the front rows and stuff. But if you take this off, this screen will raise up in the way. And so this was a student's answer to that. I don't know who put it there, but I came in one day and this was here. I don't remember even asking why it was there. I just went with it. Because it works. This is their answer to fixing the podium. There were 3 or 4 work orders put in on it. That's why we are the medical center of excellence. Actually, I think that was broken before we even became part of TRADOC. That's how long it was. That works. Things don't get fixed around this building really fast. And it's because of the unions and stuff, honestly. I once got yelled at by many people when I was on active duty because we were moving the IPAP offices up here to this floor. We used to be down lower. And the secretary's office was in shambles. I went in one day, and her furniture, she hadn't moved her, she's the only one that hadn't moved up here to her office. And I went, I mean, I got time. Why don't we just move your stuff? And she's like, well, I'm moving all the furniture too. And I said, you know, it's military. I'm like, well, great, let's do it. And I went out in the hallway and I asked a couple of students, hey, you got anything going on? Like, no, I said, you don't have to, but you mind helping me get these few desks upstairs? Just gonna throw in the elevator and go up. We did it. Oh my gosh, that caused havoc in this building. Apparently, her new office space was like 4 cubic feet short of what she must have. And so they hadn't moved it yet. And there's a whole union that moves furniture. You may not move your own furniture in this building. I didn't know that. Anyway, I had people I didn't even know coming out of the building across the way where all the admin yelling at me about taking people's jobs and all this stuff. I'm like, what? That was a long time ago, but that back in 2011. Maybe it's changed, I don't know, you might know. You still got to contract people to move stuff?
Yep.
Yeah. So,
Don't even drill holes in the wall until I do key boxes either.
no, you can't do anything. It's somebody's job. Alright, anyway. I love this place. Keeps you happy. Hey, mitosis. Let's get into serious stuff, and maybe tomorrow I can stay on topic, but you know, you never know. I try every morning. Mitosis is replication division. Oh, thank you. So I told him to raise his hand if I forgot, and I did. He was asking, remember I told y'all to remind me to tell you how I use the study picks and the self-assessment that's on each block study materials. That material, the way I built it originally, those self-assessments are not all inclusive. Meaning what I wanted people to do, and I used to not unlock them until like a day or two before the test. Now I just leave them there and I tell you what to do with them. Because the self-assessment to me was built to where you study however many slides there are, then you want to kind of gauge where you're at. You know, you want to take a little practice. You go and take the self-assessment And hopefully you're taking it, you're like, where did that come from? I never read that. Then you go back and kind of read it. It wasn't, and it gives you an idea of where you stand from your studying, because it's random questions throughout. That's how I built it. So you would realize, get a gauge of how well have I been studying. Did I pay attention to certain things? But it's not meant to be, take the self-assessment and you'll pass the test. If all you do is take the self-assessment and you think, I kicked its butt, I'll go and take the test, you're going to go down in flames. Because it's not all inclusive, right? It's examples. It's also a good way to show you info that I look at that may be more important or not. There is some good info in there. But in the self-assessment, the questions are kind of crazy, right? If you look at it, there's true false, there's lines, there's matching. That's not representative of a test. A test will only have one stem or one question with 5 multiple choice answers. There will not be true false and all that. But the self-assessment's fair game, right? It's just there to help you. The study picks are because, as I taught this more, and especially when you get into musculoskeletal, there are a lot of pictures. And there's a lot of labels. And if you know Netter's is an anatomy book, and we use a lot of their pictures, and there's always a lot of labels. So I always try to whittle down or box off what labels you need, but also it became so inclusive. Somebody once pulled out like all the pictures in a musculoskeletal and was like, there's 85 pictures, how would we, and if I have to learn to label all this, how would I know that? I tell them, well, just suck it up. No, you got it. You can go on. No. What I wound up doing is going, okay, well, let me pull out some pictures that I might use more than likely than not for the test, right? So a lot of those pictures you see, almost all, I'll be honest with you. I don't want to say every, because I might throw a random picture in there sometimes, but you will have seen it, or it will be a version. So here's this, what I'll guarantee you with the study picks. At least, we'll throw a percentage on there. I'll make it up. 90% of the pictures you see on any given testimony will come from those. It will be the same picture that's in the study book. Does that help? Yeah. On the rare chance that I have an old question that pops up that has a different version of that same picture, like it's a little to the right or the left, because people notice that stuff. But most of them are going to be your study picks. Those are there for you to learn for the test. If you come up with a question like, hey, in class, I'm looking at this study pick, and there's something labeled on here that we didn't talk about, or it's extensively labeled. First thing you do, if you see a study pick that's extensively labeled, like a Netter's picture, look at the study, or look at the picture in the slides that correlates that same picture, because it's in the slides too, that's where I got it from. And you'll see a lot of times I've boxed off what we were talking about. There may be 3 or 4 things that are boxed off that we used that picture for. That's what I would ask you to label if I was to ask you to label it. Not one of the random ones that we didn't talk about. Okay? It's just not as clean as it should be all the time, but that's how you do it. Or even better, like y'all didn't catch me in between class or as I'm walking by and say, hey, I'm looking at this study pick. What's the take home here? And I will tell you. Okay? Fair enough. That's how you use them though. I will also, at the end of only this first block, only for test 1, you're still going to ask me on test 2 if I'm going to do it, and I'm going to tell you no. Is when we first started with Canvas, a lot of people weren't good with taking Canvas tests. Like, are we going to be testing in Canvas? And you're still going to have hiccups. But I built like a practice test. What I did is I went to my test block, or my test question blocks, which I have hundreds and hundreds of them, and I just randomly pulled some questions out, looked at them and said, yeah, those will work. And I threw, I think, 20 questions, 25 questions into an actual Canvas test. They are questions that I individually wrote, and they're right out of stuff you might see like on a test. And I would give that to people so that they could, we would do it in class, we do it like it's a formal test, so we'd get some of the anxiety out. But we don't do that anymore, but I still, when we get close to the test, I will open up that practice test in Canvas so that you can take it, once again, kind of see where you're at, and you get to see questions I've written before we ever get to the first test. You get to understand how I phrase things. I think that helps people. So you will get that too. Alright, enough of that. Any questions? We're good. That's how you use them. That was a lot longer than I intended to talk about that. But if you complain about the study picks and the self-assessment and say it too much, it goes away. No, I'm kidding. I'm kidding. Okay. Mitosis. Mitosis is cell division, where we're going to look at the normal human cell. It has 46 chromosomes, and we want to take that cell and replicate it. We're going to need to replicate everything that's in that cell and all of its DNA. That is mitosis. So remember, if we have 2 chromosomes, 23 pairs, 2 in each chromosome spot, they're homologous chromosomes, which I already know, everybody's already thinking in their mind is they look alike, they have the same linear sequence of genes, and they pair during meiosis. I know that's exactly what y'all were thinking, right? No, that was some good sarcasm. I know you did. Now you realize it was on the previous slide. Somatic cells are called diploid cells because they have 46 chromosomes. When you look at a cell, I'm fixing this as I'm talking. When you look at a cell, generally cells, if they're not in mitosis, they are in what's called interphase. Okay, so during interphase, the cell is not divided. So we're going to talk about what's called the cell cycle. So if you have, let's say, a skin cell. Your skin cells are shed, if you do this and you can see it in the light, you're shedding skin cell cells. They're going all over. They're all over your house, right? Because the basal layer of your cutaneous layers here are constantly making new skin cells. I'm just calling them skin cells for now. And they move up and they slow up. Right? So those cells that are down there need to divide and make exact copies of those cells. So they're going to do what's called the cell cycle. So cells that are in like a basal layer are constantly taking one cell, making 2 daughter cells, 2 new cells, and then one will stay in what's called the cell cycle. So if a cell is not in the process of dividing into 2 cells, then it's an interphase. And what do you need to do? If I say, well, I'm trying to simplify all of mitosis before we even cover this slide. If I have a cell and it has a nucleus, it has 46 chromosomes, it has a Golgi complex, it has mitochondria, right? And I want to make a carbon copy of it. Before I can make 2 cells, if I intend to do that, what do I need to do in this cell first? It's called interphase where this happens, but what's going to go on inside that cell? I'm going to need to make a copy of the DNA. I'm going to need to, just like if you were doing it yourself, right? I'm going to need to make a copy of the mitochondria, the cytosol, all the organelles. Everything that cell has, you have to make a copy. While it's doing that, that cell's in interphase. And then it'll go into mitosis, which mitosis is the process of separating the DNA and those other cytosolic components, organelles, into those 2 cells. So we're going to build it all in a cell. So in interphase, we have G1, S, and G2. The key thing that's happening in interphase, besides making new organelles and all that, is you're going to replicate the DNA during S phase. We'll see that on the next chart. And then the mitotic phase, we're going to do nuclear division. In other words, separate that copy of DNA from the other one and do cytoplasmic division, or what's called cytokinesis, separate the cytosol and form those 2 cells. So this is what the cell cycle looks like. Right. Now I do not care how many hours this took. Right. Don't worry about the hours. But I didn't want you to think I didn't think that was important. I gotta erase it. Forget about the hours. During G phase, or or G1 phase, you're going to become metabolically active. You're going to start duplicating organelles and cytosol components. Centrosome replication begins. You're going to need centrosomes to connect chromosomes, right? Or chromatids. And then you're going to go into S phase. The only thing that occurs in S phase, and we don't care how long it took, is you're going to replicate the DNA. That cell started with 46 chromosomes, and we are going to make a copy of that. All right. Then we're going to go into G2 phase. We're going to continue or finish basically replicating the organelles, all of the products that we need, any proteins that are going to go as these cells separate, and then you will leave interphase. We're going to spend a lot more time talking about mitosis, because this discolored portion here, orange down here, is the mitotic phase, which you're going through mitosis. Okay, so the cell goes through interphase, and when it's done with that, it'll enter the mitotic phase. And underneath that, we're going to have prophase, metaphase, anaphase, and telophase. Okay, so you're taking one cell, and you're sending it, and it goes through the cell cycle. And then when it goes through mitosis, at the end of mitosis, there will be two cells. One of these cells is going to exit the cell cycle. One of the cells will go directly back into the next interphase. Right? And my lines look crazy on there, but the point is, if you take a cell, a basal stem cell, let's say, and you make a copy of it so you can replicate itself, if you sent both of those cells out to go on and become liver cells or skin cells, eventually you would run out of basal cells. So one cell always has to be in the cell cycle, and it's kicking out cells. Right? Make sense? You're kicking out exact copies. I put this on here, these lines kind of represent, and I think I need to redo them because people always forget one thing about them. When you look at this one, what would if you wanted to define this like in your mind, what am I trying to represent here? I can't get my pen right.
Go back.
Well, so I'm trying to represent what we have in DNA information. And I heard chromatid, chromosome, but really at this point, what I was looking for, and you understand this, I know, I just didn't lead you well in the question. That is a single double-stranded molecule of DNA. So that is a chromosome. Right? You're not completely wrong on the whole chromatid side just yet. I'm going to get to that. That's a chromosome. Remember the picture earlier when we had the single double-stranded molecule of DNA? There's your sequence of genes on it. I know I'm not the greatest artist, especially with the mouse, but you remember that picture? That's what that is. So that represents the cell and its 46 chromosomes, and they have condensed. All right. Now, during interphase, it's going to start out looking like that, but at the end of it, you're going to wind up with two molecules of double-stranded DNA connected in the middle by a centromere. Remember that from earlier? So this represents that we've taken this one and we made an exact copy, and now what do you call each side of that? This whole structure is a chromosome because the chromosome is a carrier of genetic information. But those 46 chromosomes have been replicated. It's an exact copy. So if you have a copy on this side, a copy on that side, but we don't want them to be just wandering off because we're about to pull these apart. They're connected with a centromere. Now what do you call each side of that? Yeah, sister chromatid, or you could say just chromatid. Sister chromatid generally means that it is identical, so that's great. They're not always identical. In meiosis, they won't be identical. So you have a copy on each side. So each side, just to reference again, is a single double-stranded molecule of DNA with a copy of a single double-stranded molecule of DNA. So you could say this, that helps people like numbers. That means there are 46 chromosomes, but how many chromatids? 92. 92. We'd say, remember, we got 46 chromosomes, we replicated, made a copy, and stuck it to it, but if you count chromatids, you have 92 chromatids. Right? Makes sense or not? Because it's kind of key to where we're going. Okay, so that's what it looks like when you replicate it. But we're gonna come back to that during meiosis, because until we get to meiosis, you're gonna think you understand that. I'll show you why later. When you look at the, yes.
So just going back to that one, where's S phase is the actual start phase, right? That's where we're beginning.
G1 is the start of interphases.
G1 is the start. Okay.
Correct. I'm doing two things at once. You know what I said about when you do that. You're doing two things terrible. The start of interphase is G1. S phase occurs between G1 and G2. That's what you're asking.
Yes, sir.
For mitosis, we got to break down. Now, under mitosis, I do want you to know what occurs in each phase of mitosis. That is a key thing. So during mitosis, you have prophase. Right? During interphase, that we just all talked about, if you were to look at your chromosomes, they are relaxed and look like cotton candy. They're called chromatin. But we need to organize them to keep things straight. So how are we going to do that? They start to condense. And so during prophase, the chromatin is going to condense into chromosomes. During prophase, the chromatin fibers are going to condense into chromosomes. I say that because I know that somebody reads that and goes, wasn't chromatin chromosomes? Yes. But think about this. I try to do this different every semester and get people on. When they study chromosomes, they can't really see them well while they're, picture it's you. You're studying chromosomes and you're looking in this cell and you're like, I know that there's a nucleus and there's all this chromatin and I'm looking at it under a microscope and I'm like, it's just a big glob mess, right? But I know that's all of their chromosomes, right? We know that. Chromatin is your chromosomes, they're just relaxed. So when you read the definition of prophase, it confuses some people because they say, well, now they can condense, the fibers condense into chromosomes. And people will come and say, well, weren't they chromosomes all along? Yes. But you were studying them, and they lined up, and they came to look like that little X. They've condensed, and so that's representative in people's minds of a chromosome. You're calling it that because now it looks like what you expect it to look like. But the genetic information was always there. So a better way to say it, I did the book way, is just say, chromatin fibers condense into a compact chromosome. That's a better way to say it, I think. All right. Metaphase. In metaphase, you're going to take the chromosomes and use the microtubules, and they're going to take all the centromeres. Remember, they're connected. Every time I get out, I got to get my finger. They're connected. The sister chromatids are connected. They have a centromere. So microtubules will attach and take those chromatid pairs and line them up along the metaphase plate. Okay, we're going to go through the definitions, and then we'll go back through with pictures. Anaphase, during that phase, you're going to take the chromatid pairs, and we're going to move them to opposite sides of the cell. You're having one cell, and it's going to start to divide. Then you're going to have to pull the DNA apart. So you're going to take this, I'm just trying to represent that X. You're going to take those two sister chromatids, line them up down the middle of the cell, and then during anaphase, you're going to pull them apart with spindle fibers on it, if you've ever studied it. You're going to pull those apart to each side. And now those chromatids would be called chromosomes. They're only called chromatids when you have a copy of that exact, or a copy of DNA attached to a centromere. and they needed to represent that somehow, so they to know that that's not just all one set of DNA. There's a copy there. So when they're connected, they call them chromatids. But when they separate them, right? I made a copy, right? I had 46 here, and I had 46 here because of what happened in S phase, and we connected them. But now we're going to line them up down the plate and separate them, to where now I have 46 here and 46 here. You see what I mean? Now they're chromosomes. They're only called chromatids when they're connected. Hopefully that links it. Yeah.
So what's the difference of the chromosomes in prophase versus the chromosomes in anaphase?
What's the difference like as
Like the look of it, the structure. Like, so it says here condensed into chromosomes, and then it says now are called chromosomes. So where's, like, what's the difference in those?
Let me show you on the picture. I got a visual. I'll show you. Remind me of that. It was coming up. Well, let's cover this slide and then I'll show you. Cytokinesis. At the end of all of this, when you get to late anaphase, remember there's anaphase and then you still have one more phase called telophase. Cytokinesis begins in late anaphase. And what cytokinesis is the division of the cell's cytoplasm. And so as you have, it starts to look like this, where you had one cell, but now you kind of have two, and you'll start to develop a cleavage furrow between them, where eventually they're going to separate through here and split into two daughter cells. All right, let's get some pictures going so we can figure this out. So during interphase, the cell is not dividing. The cell is getting ready to divide. Right? It's doing all the mission prep. It's replicating things that it needs. G1, the way to think of it is G1 and G2. G1 starts replication of organelles and things that you're going to need in the cell. G2 completes that. But in between that, you have S phase. In S phase, the only thing that occurs is the replication of that DNA. Make a copy. All right. In prophase, when you say chromatin fibers, remember I'm answering your question too, is chromatin fibers look like, in my mind, cotton candy. They're all relaxed, right? But chromatin is still a bunch of chromosomes. They're just relaxed. So most of what we're talking about is how they look. Right? So chromatins change into what we think of as chromatins. Instead of looking all cotton candy-like, they condense. They condense and start to look like this. Right? Where you have all these chromosomes and each one has two chromatids. Those would be chromatids. They're connected by the centromere. Now that we have them like that, the microtubule, down the middle of a cell is called the metaphase plate there in the dotted line, and we're going to line up all of those sister chromatids. We're going to line them up down the metaphase plate. Just like that. So basically through the centromeres. And you can actually see a good representation of an actual view of that. We're lining them up. We're getting them organized. Right? So your chromatid pairs have lined up to the metaphase plate. During anaphase, we said we're going to split them apart. So during anaphase, the chromatid pairs are going to split apart and move to opposite sides of the cell. They're going to be pulled apart. And so instead of looking like this, we're going to pull this. I don't know your names, I just have to have server. That representation there of sister chromatids is lined up in the metaphase plate, and as soon as we pull them, if we take, I'm going to draw right in the middle, we just represent one pair of sister chromatids. If we pull them apart, then we're going to pull one this way. Now there's 46 of these happening. And then we're going to pull one this way. You see what I did there? I separated them. Once I separate them, Now they're called chromosomes. So to answer your question, I'd say, well, remember back to chromosomes, this is still a single double-stranded molecule of DNA, right? Just like in the beginning. The only thing that's confusing is once we've replicated the DNA, we connected them. We made a copy on each side, so this is identical to this, and we connected them with a centromere. When we did that, they needed a way apparently to say what was represented there. So they decided to say, okay, we're going to call each side of that, because there's a copy of it, sister chromatids. So when you say that, it just represents that you have a single double-stranded molecule on one side, a copy of that on the other side, and they're connected by the middle centromere. That is what you were asking, right? No, it doesn't look like it was.
So I guess, so these chromosomes look like the one side of the X, and then when you call them chromosomes up here in prophase, they are the X. So I guess like you're calling them both chromosomes. So.
Well, that's fair. I see what you mean. Because if I was to ask you, let's just go with representing. Remember, a chromosome is a carrier of genetic information, no matter what it looks like. You got a great question on here, because some people are thinking this too. If I drew this, God, I hate that. If I did this, and I said, what would you call this? In genetics, what would you call that structure? It's a big X. Now, I didn't even, let me put a centromere there to help you confuse you. If I just said, okay, looking at this, I'm going to ask you this on a test. I put that structure up and I say, what would you call this? Chromosome. Chromosome. Okay? If I put that up on a test and I said, and I circled over here and I said, okay, there is an oval here. And I asked you, inside, in this picture, part of it, inside of the oval, what's the name for that? Chromatid. That's a chromatid because sister chromatids are identical copies connected to each other. So if you're getting that still, that's the confusing part is, okay, but we know during anaphase, we're going to separate those. Right? So if I drew this, if I drew this structure, right, and we can put it, remember they're here too. Actually, I can just circle this. What would you call that? Chromosome. You would never call what I just circled a chromatid. Because a chromatid only exists during prophase and metaphase, really. Right? Because that's the only time you have those identical copies connected. Still no. I thought
No, you're good. No, you're good.
Oh, you still had that look, and I'm like, damn, I didn't get it across.
You're good.
It makes sense. It's just a way that I think that they just had to have a way to describe what they were looking at. But
Would it might help, sir? I'm thinking that like chromatids can only exist with the centromere. Because the centromere only exists when it's holding two sister chromatids or non-sister chromatids together. Once they pull apart, there's no more centromere. It goes back to the chromosome.
yeah, I feel like I've been saying that over and over. I'm with you. I agree with you. And when you were saying that, I'm like, isn't that what I just said? No, I agree with it. I mean, did you hear what he said? The thing of it that chromatids only exist when there's a centromere, which is true. Yes.
So, like in prophase, whenever you say the chromatid condensed into the chromosome, is that like it finding the one chromosome finding this identical match, and they become like that pair, like two sister chromatids?
Yes.
That's what happens in prophase.
No, when they condense into that chromosome, it's going to look like an X, but we know that it's two sister chromatids.
Right.
Yeah. Nice. It's only this side of the room. This side of the room's all nodding like, I got it. And I'm kidding. I'm trying to start a back and forth. Are we good? I don't mind staying here because this, I mean, these are all good. I wouldn't continue talking about it. I didn't think it was important stuff to know from here and in like biochem and other classes. Because if you get confused on the nomenclature of what you're naming things, you miss simple questions. you miss something when it goes, well, that's a chromatid, not a chromosome. And you start arguing like, well, wait, isn't a chromatid really a chromosome? Because it's a carrier of genetic information. Remember when I talked about technicalities? It's how it looks during each phase. When it's connected, they're chromatids. Yes?
I was just gonna ask for like, trying to wrap my brain around the numbers, like, since it's diploid, there'd be 46, like there would be, like, before the match, and the chromosomes match, there'd be 46 chromosomes. Those would each be diploid, so there would be 92 sister chromosomes.
Correct.
And then during the phase when it separates, it would go back to 46.
Yes. Did y'all hear that? She's recapping. She's recapping what I threw out there earlier. If you have 46 chromosomes that get replicated, how many chromatids will you have? 92. Why? And this is the real take on it. Well, why? Y'all just try to make this last to the end of the hour. Why? I don't mind that at all. Why do we say 92 chromatids and not just say, well, we have 92 chromosomes? We all can divide. We know that 46 are going to each side. That would make it much simpler, right? But it doesn't remind you of something. That's the only, I wasn't there to do this. But it doesn't remind you that there's an exact copy. And we're not geneticists, because geneticists don't really talk about it the same way. They really use N, 2N, 4N, because N represents the amount of genetic information in that cell. And so if I'm at 2N, do not write this down, and do not remember this if you've already got it. So I don't want to get into this. It's representing how much genetic information is in there, regardless of what it looks like. So if you say a cell is in mitosis and it's 4N, that means we have twice the amount of genetic information a normal one of our cells has, right? Because 4N represents that you have 46 chromosomes with an exact copy. So 4N is more representative, a better way to identify that you have, 46 chromosomes genetic information. You made a copy. You didn't really add new information to that cell. You just made a copy. See what I mean? So when these fancy scientists are studying it, it helps them to keep up with it. Right? But some people love math, and saying 2N, 4N helps them even more. Or you can just look at the chart I think I put in here. It's after mitosis. We getting it or no? Somewhat. There's telophase. We got 2 new chromosomes starting to form. Now the nucleus will reform because we've already separated. We now have 46 and 46. And then they will, start to relax again. Once the nucleus reforms, the chromosomes will relax again. And then you wind up in interphase again. I'm listening to them, so we can. I'm not diving y'all out for talking. I'm saying, you got a good question?
No,
John?
we were just discussing the quantity, the numbers between 46 chromosomes, 92 chromatids,
Yeah.
and just the importance of words.
What I was trying to figure out, if you go back to, slide number 139 with the G1 phase, S phase, G2 phase. So in my head, I'm trying to make sense. How many chromosomes are in each one? So in G1 phase, what I'm understanding from what we're talking about, there's 46 chromosomes, and then it is duplicated in S phase, and then they just separate. So I'm trying to figure out, is any time where before this are there only 23? Because that 23, that means they're floating out until they get to the G1 phase. But I don't know. That's what's kind of confusing me. It's
somatic cells will never have just 23 chromosomes, right? You could say they have 23, sorry. Let's not get there yet, because that's tomorrow coming down, because that's meiosis. Don't get believe it or not, this is the easier one. Meiosis is much different. Okay, let me say this to make you, I'm going to throw you a wrench. How many chromosomes are here?
46.
How many chromosomes are here? How many chromosomes are here? 46. That's where you're getting at. That's the step you're going, wait a minute. Because you made this copy. And in your mind, you're like, well, now I have 92 chromosomes. No, you don't. You have 92 chromatids. Because they're an exact copy of each other, they don't say you have 92 chromosomes.
Okay. 46.
Which is why, and you're right on track with where I want to be, is that's why when I drew the X, remember the very beginning of genetics, I drew an X, and then I drew just one beside it, and I said, what do you call this and this? Well, they're chromosomes. So it's, a chromosome is just a carrier of genetic information, but they can look different ways. But, he's still not liking it, but during this whole time, you have 46 chromosomes. Now you copy it, you have 46 chromosomes with a copy, so you have 92 chromatids. You still have 46, 92 here, you still have 46, 92 here, but here you've split them apart, and now you have 46 and 46 chromosomes. Correct?
Sir, can you go back to slide 145? It's your interface. You give the example of the chromatid top versus the chromosome making the whole. Why is that circular part? Why is that most important?
Because it's two things. That's how chromosomes are normally look like on the chart. That's because that's a single double-stranded molecule of DNA, which is the definition of a chromosome. And you can also say, well, what is the definition of a chromosome? A carrier of genetic information, regardless of how much information it's carrying. Right? I keep repeating things different ways when y'all ask me, but that's exactly, because that's the answer, because it's a carrier of genetic information. It doesn't matter how it looks. The only confusing part that we keep circling back to is when you have a copy of that DNA and it's connected, we're going to call them chromatids. Why am I spending so much time rather than just going, if you don't got it, talk to me after class? Because if you don't get this, the naming, you will not understand meiosis tomorrow. You won't, or not tomorrow, I don't teach tomorrow. Next week. Here's the thing too. If you like the numbers, if you go, I don't know if you looked ahead, you get past meiosis, I threw this in there for the math people, where you can see during mitosis and meiosis, the number of chromosomes per chromosome counts. And I promise you this, we're going to go into meiosis next, Monday, probably? And we will recap mitosis, we'll go into meiosis, and then we'll sit there and hover on these two until we get them, because they're not that difficult, and it's not something you need to be missing. It's because we're not getting that deep into genetics. What we're getting tied up on is naming things, right? You don't want to miss something like that. Yeah.
All right, sir. So this kind of goes back to the last question. So like during anaphase there, it says 92 chromosomes.
It's representative, but
So they're really chromatids, but 46 chromosomes when they're connected at the centromere.
yeah.
Okay, because that's what I was confused about. We're calling them chromosomes, but they're really chromatids of a chromosome pair.
They're really 92 out here, right? But the reason this chart is doing that is, I can answer your question, that's good. Now I just figured out what you asked me. That says 92 because we've already pulled them apart.
Oh, so they're saying the two cells. So the two cells. Okay.
Yeah. Does that make sense?
That makes sense.
Now that we split them, you would have 92 chromosomes. Is everybody, that's a great, I'm glad you mentioned that. Is that great? You see that over here? Y'all, they're all acting like they've got it compared to y'all. That's not fair. No. You see what he was asking? You're like, well, wait a minute. You keep telling me during interphase and all this and prophase that we have 46 chromosomes, 92 chromatids. Then you came in here and look at it and say, well, why does it say 92 here? Because we already separated. Remember what I said, once you separate the chromatids, automatically now you call this chromosomes. I'm really glad you asked that, because I think I see a lot more head nods now going, okay. If not, just fake it until next week and we'll figure it out. Okay? I gotta let y'all go. Y'all have a good day. I'll see you.